Today we're going to start Unit 8, and that's on evolution and natural selection. This is going to be our last unit that we cover in Biology 101. So looking here, this is going to be a glass-winged butterfly. And with this glass-winged butterfly, notice that it has developed what look like their eyes on the back of its wings. And so this is going to be an evolutionary adaptation to this insect, so those who might be predators won't actually know what side's going to be the front or what side's going to be the back of this animal. So give your opinion. The human population is evolving. So evolution in action. We can actually see evolution occur right before our eyes. But before we start, the first thing you need to understand is going to be population. So population is simply going to be a group of organisms of the same species that are living in a particular geographic region. They could be small or they could be large. For example, we can have those that are existing in a test tube state, or they could live in an actual state. And so different types of organisms, depending on where they are, can live in a certain area, and that's going to be its population. Now look at this question. Could you breed fruit flies that could live longer than 20 hours on average? Well, by looking at this chart, this is going to give you the average amount of time that a fruit fly can live without any type of food source. And so we see here the average is going to be right at along 20 minutes. And so the answer usually is just under a day, that 20 hours, give or take a few hours, because you see some do live past that, but some do die before. So in order to be able to see if we can do this, we're going to conduct an experiment. So with this experiment, first we're going to start with a population of 5,000 fruit flies. In biology, the term population, like we said, has a specific meaning. It's going to be a group of organisms, remember, of the same species living in a particular area. In this case, it's going to be this large container. And so that's where they're going to put them, this cage. And so that is a population. We have 5,000 there. So with those, we're going to remove the food. And after the 20-hour period, we're going to have about 80, or actually we're going to wait till about 80% of those have died. And then we reintroduce the food. And then they can eat. Once we do that, the ones who are feeding again will then breed. And once they breed and lay eggs, then those should be born, and they should actually carry the gene that helps them live longer. And so looking here, we can see that now you've started that new generation. It consisted only of the offspring of those fruit flies that were able to survive without food for the longest amount of time. We did up to 80% have passed, so this is the 20% that lived. And when these eggs hatch, the flies show an increased resistance to starvation. The average fly in the new generation can live for about 23 hours compared to the 20 hours in the original population without food. And again, some of the new fruit flies will survive more than 23 hours, and of course you will have some that survive less. So what if you kept repeating these three steps? So with each new generation, what you're going to see is a slight increase of the amount of time that they can live without food. And so over time, their survivability rate is going to increase. Eventually, over many, many generations, after about 5, what would you expect looking at that? So after about 5 generations, you'd expect it to survive about 32 hours. But after 60 generations of allowing those only the top 20% to survive and then feeding them and allowing them to reproduce, you're going to see them being able to live up to 160 hours. And of course, you're going to have give or take. Some are going to live a little bit less and some will live a little bit longer. So what happened here? Well, evolution happened. There was a genetic change in the population of fruit flies that were living in the cage. And so every time the fly in generation 60 population, even the fly with the worst starvation resistance, is still going to be more than 7 times better at resisting starvation than the best fly in the original population. So here, this evolution is going to be a result of what we refer to as natural selection. So we'll discuss natural selection in more detail later, but it is the consequence of certain individual organisms in a population being born with characteristics that are enabling them to survive better and reproduce more with the offspring of other individuals. And so each time we increase that by 20%. So looking here, does evolution occur? Well, it's going to be a astounding yes. We do see evolution occur. This experiment actually helped answer our question with that. So we can watch it happen in the lab whenever we want. Now remember our discussion about scientific method for an experiment results invalid. They must be reproducible. And so if you were able to do an experiment, or we saw this fruit fly, you should actually be able to take these, put them in a container, and repeat this experiment yourself and still come up with close to the same conclusions. All right, so with dogs, we actually see this happening because lots of people nowadays, they want those small, tiny teacup dogs. And so with that, they only allow the smallest dogs in each litter to reproduce. When they do that, the outcome is going to be small dogs. Well, every time they take the smallest and the smallest dog, it's going to decrease over time to where we've taken a dog from an average size, and now we've created a miniature version of that animal. Can we do that with rabbits? Of course. In one case, we allow only the fastest rabbits to reproduce, and then as we do that, that makes each generation after that a little bit faster and a little bit faster. So with that case, you could actually see that in a natural population if they were being hunted by things such as foxes. So only those that could outrun the foxes or who were the fastest runners tend to survive. With those that tend to survive, they reproduce, and their offspring should be fast as well. Now, of course, you're going to have some that aren't quite as fast, and those, of course, might be preyed upon by the foxes, but those who did survive are probably going to be the ones who got the gene for running faster. All right, evolution. What will you learn in this section? Well, you should learn how does evolution occur? You should learn what types of changes can evolution cause in a population. You're going to learn 5 primary lines of evidence and evolution by natural selection. So looking here, what was the control for the starvation-resistant experiment? The answer is going to be number 1. So we're actually using generation 0. This is not the first generation that we created. So looking at that, you should be able to see that by using this chart right here. So in summary, the characteristics of individuals present in a population can change over time. We can observe such changes in nature, and we can even cause it to occur. So Darwin journeyed to a new idea. So Darwin, of course, there's a lot of talk about him because he made a journey to what's known as the Galapagos Islands. Here we see the Galapagos land iguana, and that's going to be one of the species that he was able to observe when he made his travels. So Darwin's journey to an idea. Charles Darwin is going to grow up in an orderly world. He's growing up during Victorian British society, where he is going to inhabit specific beliefs. They're going to be very religious during this time, and they also only believe that Earth is around 6,000 years old. And so that's not going to explain what some scientists are starting to find with specific fossil records. They're starting to uncover different records of species that no longer exist. And so this is going to leave a lot of people wondering what has happened. So with this, like we said, he grew up in Victorian Britain. He's going to have little change to scientific belief for the past 200 years. And so from the 16 to the 1800s, not a lot's happened. They do rely heavily on biblical explanations, and to them, they were sufficient to explain these natural events. So most people are going to think that Earth's only going to be around 6,000 years old. They also believe that Earth was unchanging. But as he travels to these different places in his 5-year journey, he's going to find out that Earth is changing dramatically through natural events. So organisms also here, they believed, were put here in groups called species or kinds. And then they think that each species was created individually, and those species did not change, and they never died out or went extinct. So looking here, we see that Darwin wasn't the only one to shake up the world. We have people here such as Buffon in 1778 who suggested that the Earth was about 75,000 years old. Of course, that didn't go over very well. Then we have Cuvier in the 1790s who's going to explore coal and slate mines, and he's going to start to find fossil remains that were unlike any living species that we have seen on Earth. So here are some of the things that they discovered in these mines. They discovered the mastodon, the Irish elk, and giant ground sloth. So if you compare those to the average human being at the time, then you see how much larger these things are. Now we do have smaller versions of these organisms now, but they are nowhere near the size of the ones that were found in the fossils. So these animals must have gone extinct. Jean-Baptiste Lamarck is going to be a biologist in the early 1800s. He's the one who puts forth the idea that living species might change over time. And so with these species, he said that not only was it starting to appear that species could disappear from the face of the earth, but they could change and actually change over a period of time into a completely different looking type of species. So all those his ideas about the mechanisms by which this might occur, they were wrong when they went back and looked at them. He still had the right idea about what was going on. So his willingness to question sacred truths actually helped him ask questions and helped others to be able to open up this study later on. Moving on, we next have Charles Lyell. Charles Lyell was a geologist. In 1830, he writes a book called The Principles of Geology. Here he states that geological forces had shaped the earth and were continuing to do so. These are going to produce things such as mountains, valleys, cliffs, and even canyons. He said it's a gradual but constant change of Earth. And so some of the things that are going to actually do this, he thinks, are going to be things such as earthquakes. They're going to be things such as volcanoes. They could be large storms, tsunamis, and any type of what we would consider to be a natural disaster can come in and actually change things over time. But also, you might have certain things changing, such as the tide coming in and out, or withering away or weathering of rock. So looking at this one, which idea suggests to Darwin and others that living things might change over time? The correct answer is 4, which is both 2 and 3. So, a job around the world survey ship is going to allow Darwin to indulge his love of nature and to make observations that enable him to develop a theory of evolution. So at the age of 16, Darwin's going to go to the University of Edinburgh to study medicine, following his father's footsteps. But he was bored. He didn't like medicine. And so he's going to leave at the end of his second year. That just wasn't his thing, he said. Darwin then pursued an education in the ministry. And so he's going to study theology at the University of Cambridge. Shortly after he graduated with his theology degree in 1831, Darwin's going to land what's known as his dream job. He gets a position as a gentleman's companion. And this is going to be for the captain of the HMS Beagle on a 5-year round-the-world expedition. So he gets to go after his real love, which is going to be nature. So he becomes what's known as a naturalist. So here we have him riding the HMS Beagle, and it's going to be a 90-foot sailboat. And he's going to go on this 5-year journey around the world, surveying different islands and different land masses that he comes to. And so, like we said, this is going to be a dream job for Darwin because he wasn't really into the ministry. And, of course, we knew that he didn't really want to go into medicine, which he started out like his father did. And so looking at this picture, we can see where Darwin traveled. To avoid seasickness, Darwin spent as much time as possible on shore. So he did not stay on the ship like some did. He's going to actually exit and go explore. It was there that he's going to find in his field work his true calling. At each stop, he would eagerly investigate the new worlds that he found. In Brazil, he was enthralled by the tropical forests. In Patagonia, he explored beaches and cliffs, finding spectacular fossils from huge extinct mammals. Elsewhere, he explored the coral reefs and barnacles, always picking up specimens for museums and recording his observations so he could use those later. The only book Darwin took with him on the Beagle is going to be Lyell's Principle of Geography. So the idea that Earth was an ever-changing planet would serve Darwin well when he's sitting here thinking his first few years during the journey on the Beagle. And once he stops in the Galapagos Islands off the northwest coast of South America, this is where he's really going to take this book and put it to good use. So he uses this group of volcanic islands in the Galapagos, which is going to be home to many unusual species, from things like giant tortoises to extremely docile lizards, which he's going to use little effort to run away from him. So he actually has to try to avoid stepping on these animals. And so they're very, very tame, and he's not used to that. So Darwin's going to be particularly intrigued by the wide variety of birds also when he's there, especially the finches, which seem to be dramatically different from island to island as he went to the different locations. So here you see those Galapagos Islands, and notice how each one of them looks different. Here he's going to find things such as those giant tortoises. You'll see those on the bottom. We saw the lizard earlier. And then he sees a wide variety of the finches that he's going to look at. So some things that he's going to notice. So first, it's going to be involved with the finches that he is going to collect. He notes the different physical characteristics or traits, such as body size, beak shape, and feather color. Also, he notes that they resemble a single living finch that's found on the mainland of Ecuador. Darwin also thought that maybe finches on the island used to be part of the same mainland population. And he's going to separate and divert. He thought that they separated and emerged from the original population gradually and formed a new population of a similar species. So he thought that resemblance did seem suspicious, and perhaps that those finches resembled the mainland because that's where they came from. And so after being separated from the mainland over a period of time, they started to change over to what was actually on the island that they were located on. So if that island had, let's say, sharps, or not sharp, but hard seeds, then they might need a thicker beak that was able to crack those seeds. If their food was found in a small space, they might need more of a pointy beak in order to get that food out. Okay, next, at every location, there's going to be a striking similarity between the fossils of extinct species and living species of the same area. For example, in Argentina, he found giant fossils from a group of organisms called glyptodonts. The extinct glyptodonts are going to look like armadillos, a species that still flourished in the same area, but armadillos only weigh about 10 pounds. The glyptodonts are going to be giants. They're going to be about 10 feet long and more than 4,000 pounds. So there's a huge difference in these there. However, he noticed that between the fossils and the current species, they looked alike. So he's going to deduce that the glyptodonts resembled the armadillos because they're actually going to be ancient relatives. Inspiration's going to strike Darwin when he reads, for amusement, the essay on the principles of population by the economist Thomas Malthus. Darwin's going to speculate that rather than the future holding certain catastrophes for all, maybe the best individuals are actually going to be the ones, and it ensures the struggle for existence, and the worst would lose. So they're going to lose out, and it's kind of that survival of the fittest thought. And so if so, he suddenly saw that favorable variations are going to tend to be preserved and unfavorable ones are going to be destroyed. And so here we see a picture of Thomas Malthus. So next we go to Darwin's book. Basically in 1842, Darwin's finally going to be prepared to release his first draft on his ideas, which is going to be a 35-page paper. He also had an inkling that his ideas would rock the world. So he's kind of worried about releasing this book because he doesn't really want to, number one, go against the church. Number two, he doesn't want to be ostracized by society. So inexplicably, he's going to put his sketch into a drawer where it remained for 14 years, despite repeated warnings from friends that he should go ahead and publish his research before someone else arrived at the same conclusion independently. And so this is exactly what's going to happen. Unfortunately, he had to rush and publish it in 1859, and he gave it the name Origin of Species. But he wasn't ready to publish. But once he found out that somebody else had come up with that idea, he knew that he had to. So this is where we're going to come to our next big player in the theory of evolution. Alfred Russel Wallace is going to arrive on the scene. So in 1858, in a letter to Darwin, he's going to lay out a clear description of a process of evolution by natural selection. And he asked Darwin to publish if you think it's worthy. Once Darwin read this, though, he is going to be crushed because he thinks that he's the only one who has come up with this. He traveled around the world. He actually saw the different animals. He saw the fossils. He saw how things were changing over time. And so he thinks all my originality will be smashed. And that's a quote that he was quoted saying in a book. So they arranged for a joint presentation to be given. And then once they gave that, the result was that both Darwin and Wallace were going to be credited for the first description of evolution by natural selection. Now, it was thought that Russel Wallace, and here's a picture of him, came up with this idea when he had a high fever. They said he was in the fits of malaria. And so he just came up with these ideas. They kind of came to him while he was sick, and so he wrote these ideas down. And he knew that Darwin was looking into this, but he wasn't ready to publish himself. So that's why he sent it to Darwin for him to read through. And so looking here, we have the evolving beliefs of Darwin. Before, they thought all organisms were put on Earth by a creator at the same time. Organisms were fixed, there were no new organisms, and nothing ever went extinct. Also, they thought the Earth was about 6,000 years old. By 1859, when they first published their Origin of Species by Darwin, now we're going to look at how things start to change. Organisms change over time is a new idea. Some organisms have gone extinct. Earth, they know now, or think, is going to be more than 6,000 years old, and the geology of Earth is not constant, but always changing. So looking here, all the statements below are going to be true except the correct answer is going to be number 4. So 4 mechanisms can give rise to evolution. So close up of the forewing of a green tiger beetle here. The coloration may help deter predators by resembling a species of wasp with a harmful sting. So this is going to be a close-up look at that. So evolution is going to occur when the allele frequencies in that population change. So looking here, suppose that you were put in charge of a population of tigers at a zoo. Most of those tigers are going to be orange with black stripes, but every now and then, sometimes you get an occasional white tiger. Now that tiger is going to have the albinism gene. We talked about that when we talked about Punnett squares. And so white tigers, we know, along with most albino animals, are going to draw more visitors. They draw more of an attraction. So how would you increase the population of white tigers in the zoo? Well, here you can use your what you know about populations, what you know about genes, and you can help change these over time. So you're going to alter the population. You're going to increase the white phenotype through a breeding program. And so with this, as the generations go by, you're going to start to have higher proportions of white tigers. And so this evolution is going to be the change on allele frequencies of the population. And that's what you will be doing. So alter the population. You're going to increase the white phenotype by acquiring them from another zoo possibly and add them to your population, or remove orange tigers from your population. Individuals themselves do not evolve. You have to evolve as a population. So what you need to do is you need to go in and you need to make sure that the alleles that you're looking for, let's say you change the proportion of white fur pigmented alleles in that population, and an individual tiger, of course, has 2 alleles, just like we talked about with fur and other genes within that population, and you're going to go after that white allele. So it's going to be helpful to think of each allele as having some part of like a market share of all of these allele. So in the tiger example, the white allele initially had little market share, perhaps as small as 1%, but over time, though, they're going to become a larger makeup of, or a larger proportion than the total pigmented alleles. So you think, well, why would the white not be so prevalent? Well, because that would make them stick out in their environment versus the ones that were orange. So here, looking at this one, you see evolution changing throughout that population. So we started out with the first pie chart, and we see that that proportion is going to be a major orange fur pigment for those alleles. And then over time after breeding, we're going to increase the amount of white fur tigers and decrease the amount of the orange fur. So 4 evolutionary mechanisms that are actually going to help change things over time or change these allele frequencies. The first one's going to be a mutation. That's an alteration of base pair sequence in DNA of an individual's gamete-producing cells that change an allele's frequency. So it has to occur in the gamete cells. So it has to happen in the sperm or in the egg. If it happens in a skin cell, that's just going to be with that individual, and it's not going to get passed on through other generations. Also, there's what's referred to as genetic drift. So that's going to be our second evolutionary mechanism. And that's simply going to be a random change in allele frequencies unrelated to any of the alleles' influences on reproductive success. Next, we have migration. That's a change in allele frequencies caused by the individuals moving into or out of a population. And number 4, we have natural selection. That's a change in allele frequencies that occurs when individuals with one version of a heritable trait have a greater reproductive success than individuals with a different version of that trait. So now looking at this one, which choice below would not lead to a change in frequency of alleles present in a population? The answer to that is going to be number 4, the artist painting the shells of a population of hermit crabs. Mutation. This is going to be a direct change in DNA of an individual, and it's going to be the ultimate source of genetic variation. So here, it's going to be an alteration of a base pair sequence. And remember, when we talked about DNA in Unit 5, we talked about how we have the A's and T's and C's and G's. This is going to be when those do not get copied properly, and so they're going to mutate, and we end up with the wrong set of instructions there. So when this alternation occurs in a gene, it's going to change the DNA sequence, and that change could possibly change that allele, or what's being produced. So here, say for example, that the mutation changes one of the 2 blue-eyed alleles that an individual possesses into a brown-eye allele. If this occurs in the sperm or egg-producing cells, which are your gametes, it can be passed on to the next generation. The offspring then may be born carrying the brown-eye allele. When this happens, the proportion of blue-eyed alleles in that population, for example, their market share, is going to be slightly reduced. And so when the market share of a brown-eyed allele is then going to be slightly increased. Here, evolution has occurred. All right, so which are inherited? Somatic cells or reproductive cells? Where would mutations get passed on? If you answered reproductive cells, then you are correct. So what causes mutations? Well, the processes of cell dividing can go wrong in several different ways. It could happen during the process of mitosis, or it could happen during meiosis. And there's also environmental factors that can cause these genetic mutations. You could have things such as radiation, or you can have chemicals. But any way you look at it, mutations are going to be random. So with this randomness, we cannot predict ahead of time which individuals are going to have which mutations, and we cannot predict whether the consequences of that mutation are going to be benign, which means that it doesn't help or hurt, whether they'll be harmful, or whether they'll be helpful or useful. So here, in addition to being a mechanism of evolutionary change, mutation has another more important role relevant to all the mechanisms of evolution. And that's going to be including natural selection. So mutation is going to be the ultimate source of genetic variation in a population. So this is what's going to give us mainly all of our differences. We saw that a mutation may lead to the conversion of one allele to another that is already found within the population, as for example, blue eyes and brown eyes. More importantly, though, these mutations may create completely novel alleles or ones that never occurred before. So this could be a new protein, a new color, or a new trait. For this reason, mutations are going to be critical to natural selection. So all variation, the raw material for natural selection, must initially come from mutations. Despite the vital role of generation of variation, nearly all mutations, unfortunately, are either going to have no impact on an organism's fitness, or they have a negative impact on that organism's fitness. So they can cause things such as an early death, or they can reduce the organism's reproductive success. So mutations are to normally functioning alleles that code for normal functioning proteins are going to typically result in either an allele that codes for some protein or a new allele for a non-functioning protein. So it messes up what was occurring or happening properly already. The latter case, almost inevitably, is going to reduce an organism's fitness. For this reason, the bodies protect our sperm or egg-producing DNA with a variety of built-in error-correcting mechanisms. And so with these mechanisms, we're going to see as a result these mutations will be fairly rare in humans in order of about 1 mutation in every 30 million base pairs in each generation. Okay, so looking at this one. When you read this, mutations are acted upon by natural selection only. The correct answer is 2, when found in germline cells. And so germ cells is going to be another name for gametes. Genetic drift. This is going to be a random change of allele frequencies in a population. So looking here, imagine that in a population, there are going to be 2 alleles present for a particular trait, such as the cleft chin. It's going to be a dominant trait, so individuals with either 1 or 2 copies of this dominant allele exhibit the cleft chin. Now, suppose that 2 heterozygous people have a child, and you've done the Punnett squares. Which combination of alleles will the child receive? It's going to be impossible to predict because it's going to depend completely upon which sperm fertilizes which egg. So basically, it's going to be the luck of the draw. If their sole child inherited a recessive allele from each parent, would that population allele frequency be different? Yes, it is. After all, there is now another individual in that population, and that individual has 2 recessive alleles. And so there's slightly more recessive alleles in that population now. And because a change in allele frequency has occurred, evolution has occurred. It's going to be equally likely that this couple's only child would have received 2 of the dominant alleles rather than the recessive alleles. But in either case, because a change in allele frequencies has occurred, evolution has happened. All right, looking at this next slide. So we see in small populations, we'll see this change in allele frequencies, and this is going to be referred to as the genetic drift. And basically, going back to a genetic drift, it's going to be a random change in allele frequencies in a population. With this, you're going to have fixation, and that's going to be when an allele's frequency becomes 100% in a population. And then the genetic drift is going to reduce genetic variation in that population. And so the important factor that distinguishes genetic drift from natural selection is going to be the change in allele frequencies not related to the allele's influence on its reproductive success. 2 special cases of genetic drift in a population is going to be one called the founder effect and then one called the population bottleneck. And these are both going to be important in evolution of populations. So going on to the founder effect, you're going to have a small number of individuals that may leave a population and become the founding members of a new isolated population. The founders may have different allele frequencies than the original source population, particularly if they're going to be of a small sample group. So then that leads us to the question, why are Amish people more likely to have an extra finger? Well, the Amish population in the United States is going to be believed to have been established by a small number of founders. They carried an allele for polydactyly. This is going to be a condition of having extra fingers and toes. So as a consequence, today this trait, while still kind of rare, is going to occur much more frequently among the Amish than among the rest of the United States population. And so here is a picture of polydactyly. Sometimes it's not going to be as extreme. Sometimes you might start to see just a little nub coming off the side. And sometimes you see the entire extra digit. Population bottlenecks. So occasionally a famine, a disease, or rapid environmental change can cause deaths of a large population, sometimes as much as 90% or more of that population. So because the population is going to be reduced to a small fraction of its original size, it's going to reduce and be called the bottleneck effect. Example of this is occurring in the cheetah population about 10,000 years ago at the end of the last ice age. A cataclysm or overhunting occurred, and nearly all cheetahs can be traced back to about a dozen lucky survivors after this happened. And so here you have it. So here, looking at this one, the American bison is going to be hunted into near extinction by early settlers. Which answer is correct about the current population of the American bison? Well, the answer is going to be number 1. It has experienced a population bottleneck. So only those that did survive are going to be the ones that carried their traits on. So the variation in bison now is going to be a lot less than it was before they were hunted to almost extinction. Migration into or out of a population can change allele frequencies. So the third mechanism, like we said, is going to be evolutionary change. Migration is also going to be referred to as gene flow. And so it's the movement of some individuals or species from one population to another. And so here, this movement from population to population within a species is going to distinguish migration from the founder effect, in which the individuals are going to migrate to a new habitat previously unpopulated by that species. So if the migrating individuals can survive and reproduce in the new population and also carry a different proportion of alleles than the individuals in their new home, then the recipient population experiences a change in allele frequencies and, of course, consequently, is going to experience evolution. And because alleles are going to be simultaneously lost in the initial population, that population too will experience a change in allele frequencies. So looking at this one, we see migrating birds. And so we have two populations there of these migrating birds. They're going to be on each side of this mountain. As some move or migrate from one side of the mountain to the other, we see how their alleles and the ones that they consist of move over. The population too is going to increase for the green alleles, and it takes on some more of the blue. But because that section was lost from the other side of the mountain, you're going to see how now it has less of a market share of whatever trait the green is representing. So with gene flow, that's going to be migration. That's going to lead to a change in allele frequencies in a population as an individual moves into or out of a population. Which choice below is going to be an example of migration? Well, the correct answer is going to be number 4. When 3 simple conditions are going to be satisfied, evolution by natural selection is occurring. And so there must be a variation for a trait within a population. That variation must be inheritable, and those individuals with one version of the trait must produce more offspring than those with a different version of that trait. So with condition 1, we see variation of a trait. Variation is not going to be limited to physical features such as fur color or face shape. The organism can vary in physiological or even biochemical ways too. For example, one population might be allergic to milk or lactose intolerant. Another population might have sickle cell. So they might be biochemically or physiologically different, and you can't see that from the outside. Behavior variation is going to be from things such as temperament to learning abilities to interpersonal skills. Condition 2, heritability. We can call the transmission of traits from parent to their children through genetic information inheritance or heritability. And you can see that here in this picture with Goldie Hawn and her daughter. You can see specific traits that they have in common, so her daughter inherited those traits from her. And condition 3, differential reproductive success. So Darwin derived that a third condition of natural selection from 3 fairly simple observations that he made. There are more organisms born than survive. Number 2, organisms are going to continually struggle for existence. And number 3, some organisms are going to be more likely to win this struggle and survive and reproduce. Differential reproductive success. So, This three-part observation, which goes by the complex name differential reproductive success, led Darwin to his third condition for natural selection to occur. From all variations existing in a population, individuals with traits most suited to reproduction in their environment generally leave more offspring than individuals with other traits. Here in this picture, we see that the tiniest dog in this litter is going to have a reduced differential reproductive success. Its more robust siblings are going to prevent access to food that it's going to need to grow and thrive. Also, when they play around, they might play around more rougher with the runt because it is less likely to play back. Also, another example is going to be the experiment that we talked about in selecting for fruit flies that can survive for a long period of time without food. There we saw that the fruit flies that inherit the ability to pack on the fat when the food is available end up leaving more offspring than those inheriting a poor ability to actually pad their little fruit fly body frames, and with less fat deposits. So those portly fruit flies are going to have a greater reproductive success than the ones that weren't able to keep the fat on. So here, natural selection causes evolution as individuals with one version of a heritable trait have a greater reproductive success than individuals with different versions of that trait. And see here, the first, number one, we're going to see the different variations of a trait. And so all of those different colors are going to be different alleles or versions. 2, it has to be inherited, so it has to be able to be passed from parent to offspring. And 3, it has to have a good differential reproductive success. So those individuals with the version of that trait need to have more offspring than the other traits. So natural selection basically here is going to be when these 3 conditions are going to be satisfied, the population's allele frequencies are going to change, and consequently, evolution by natural selection has occurred. Fruit flies can live between 15 to 38 hours without food. The range of survival times fulfills the condition necessary for natural selection. Which one do you believe? The answer is going to be 1, variation in a trait. So most agricultural pests are going to evolve resistance to pesticides. How is that going to happen? So basically, another way of looking at natural selection is to focus not on the winners or individuals who produce more offspring, but on the losers. So natural selection can be turned on its head and viewed as an elimination of some heritable traits from a population. So with this, we can look at why are these pests disappearing? Well, because they're susceptible to the pesticides. Why are they susceptible to the pesticides? Because they don't have the allele to live with that toxic substance. So the ones that are left are the ones that can resist it. And so here we see those 3 again. We have the variation of the trait. So we have 3 different speeds of rabbits. We have slow, a medium, and a fast. And then we have that fox. So the fox is going to be more likely to eat or prey on the slow and medium runners. However, the fastest runners are going to be able to outrun the foxes, and so their children, or they are going to be able to reproduce, and their children will survive. Next, which choice below would explain insect resistance in pesticides? Well, the correct answer is number 4, all of the above. So you have a trait that doesn't decrease in frequency simply because it is recessive. And so with this, we can actually use things such as Punnett squares to figure out how these different traits will be found in a population. And for example, just because it's a dominant allele doesn't mean you're going to see it more. And just because it's a recessive allele doesn't mean that you're actually going to see it less. And so this is going to be shown through a Hardy-Weinberg law. And so that's basically P plus Q equals 1. A good example of it, one dominant trait is going to be for achondroplasia. Achondroplasia is going to be dwarfism. That is actually, like we said, a dominant trait. But do you see as many people with achondroplasia in public as you do of normal height? No. Normal height is a recessive trait. However, you do tend to see that one more frequently. And so that's an example of not always seeing the recessive as the least prevalent. So you can go through those. So with the Hardy-Weinberg equilibrium, basically it's going to apply when there is no natural selection, no mutations, no migration, and no genetic drift, and random mating. So basically, in summary, if we know the frequency of each allele in a population, we can predict the genotypes and phenotypes that we should see in that population. Also, the phenotypic frequencies in a population are not going to be those predicted from the allele frequencies. That population is not in the Hardy-Weinberg equilibrium because an assumption has been violated. Also, either non-random mating or evolution is occurring. But as long as the Hardy-Weinberg assumptions are not violated, recessive alleles and dominant alleles do not change their frequencies over time. So through natural selection, populations of organisms can become adapted to their environment. Here you're going to see a harp seal with its white coat, and it's going to actually help hide it in the snow from polar bears. So it's adapted over time. Those that would have a darker coloring would be more visible, and therefore they would be hunted by those polar bears and less likely to survive or to reproduce and have their offspring survive. Right. So traits causing some individuals to have more offspring than others become more prevalent in the population. Reproductive success is often going to be referred to as far as a fitness. It's going to be a measure of relative amount of reproduction an individual has with a particular phenotype compared to those reproductive outputs of individuals of the same species with an alternate phenotype. So basically what this is saying, those who have specific traits and reproduce more are going to be considered more fit. So if you have more children, evolutionarily speaking, you are a more fit individual of that species than somebody who has less offspring or children. The fruit fly example. One fly is going to carry the genes for a version of a trait that allows it to survive a long time without food. The other fly has genes for a different version of that trait that allows it to survive only a short while without food. Which fly has the greater fitness? It's going to be the one that's able to reproduce with the most offspring, and those offspring able to reproduce. Now in that case, if there's no food, then it's going to be the one who's able to live the longest. But if there's plenty of a food source, then and the ones who don't live as long without food, but they don't have to go without food, then it could possibly be the ones that can go the shortest time without food. Just depends. So the alleles carried by the individual with high fitness will increase their market share in a population over time, and that population will evolve or change. So one fly carries the gene for a version of a trait that allows it to survive. The other one didn't, and so we talked about that. There's going to be 3 important elements to our organism's fitness. It's going to be measured by other genotypes or phenotypes in its population. Fitness is also going to depend on specific environment in which that organism lives in. So looking at these mice, that's just going to be different fur color. If the mice were in a lighter habitat, then the lighter mouse will survive because it's less likely to be seen on the ground, and so it's less likely to be eaten by a predator. However, if you move them to a darker background, the white or light colored mouse is going to be more likely to be seen and eaten by a predator than the darker mouse. So it's just going to depend on its environment. And third, fitness depends on the organism's reproductive success compared to other organisms. So it may have the better trait, but if it does not reproduce, then it's not considered to be as successful evolutionarily as those who have more offspring. Because if you don't reproduce, you cannot pass those good genes that you have on to your offspring or on at all. So, survival of the fittest. Is this a misnomer? Well, survival of the fittest is going to be a misleading phrase since it's individuals that have the greatest reproductive output that are the most fit in a population. So it becomes more of a meaningful phrase if we consider a description of the fact that the alleles that increase an individual's fitness will help them survive in a population more than alleles that decrease that individual's fitness. So with these, they're not actually going to be related to the health and fitness directly. It's simply going to be those who have more offspring. So looking here, which one do you think it is? It is going to be number 5. So an organism in a population can become better matched to its environment through natural selection. So as Darwin noted over and over again during his travels, organisms that possess traits that allow them to better exploit their environment in which they live will tend to produce more offspring. And then, since it tends to produce more offspring, then that offspring will carry their alleles. Here's where adaptation comes in. Adaptation is going to refer to both the process by which organisms become, 1, better matched to their environment, and 2, specific features that make that organism more fit. So some examples. We have bats that have extremely accurate type of hearing called echolocation. And so for navigation, they're going to use this echolocation to help them find food, to help them navigate their way through the skies, and to help them actually go back to where they came from and be able to land properly so they can pick up on different things that are around. Porcupine quills are also going to help porcupines keep away predators. They are able to fend them off, and they can actually even cause these quills to kind of shoot off and into a predator before it reaches it so it doesn't become harmed at all. So moving past the catalyst. Going to natural selection. So natural selection does not lead to perfect organisms. If the least fit individuals are going to be continuously weeded out of the population, we might logically conclude that eventually fitness will reach a maximum and all organisms in that population are going to be perfectly adapted to their environment. But unfortunately, this never happens. So here, consider one of the many clearly documented cases of evolution occurring, that of the beak size of the Galapagos finches. Over the course of a multi-decade study, the biologists Rosemary and Peter Grant are going to closely monitor the average size of a finch's beak. They found that the average beak size is going to fluctuate according to what type of food supply that's available. So during dry years, when the finches had to eat large, hard seeds, bigger, stronger beaks are going to become the most common or the norm. During wetter years, you'll see smaller beaks that are going to be more successful since they're able to have a surplus of that small, soft seed. So the ever-changing average beak of that finch is going to basically just be dependent upon what environment they're in at the time and what's going to work the best. So evolution in general and natural selection specifically do not guide organisms toward betterness or perfection. If the environment changes, the alleles causing the traits favored by natural selection may also change too. So why doesn't natural selection lead to the production of a perfect organism? Well, factors that prevent them from progressing to inevitably towards perfection. We have environments that change quickly, so it might have been the bigger, tougher beak that was awesome, but then we end up with a climate change and we have a lot of rain, and so now that beak is not necessarily needed. Variation is going to be needed as raw materials for selection because once that wetter climate comes in, they're going to then need the smaller beak. There also may be multiple different alleles for a trait, and so each one's going to cause an individual to have the same fitness. So in summary, natural selection does not lead to an organism's perfectly adapted to their environment because environments change, mutation doesn't produce all possible alleles, and there's not always a single optimum adaptation for a given environment. All right, so let's review the following figure and then answer the two questions that follow. So this was when we were talking about the dry weather and the harder seeds, we needed a tougher beak, and then the wet years, we had the finer seeds that were softer and the smaller beak. Now, given the beak size in mature adults does not change, what factor is driving the change in average beak size of the Galapagos finches over time? The answer is going to be number 4, both 2 and 3. What would you predict about beak size if a Galapagos experiences a wet year? The answer is going to be number 1. More birds with small beaks will survive. So artificial selection is going to be a special case of natural selection. So this is going to be the process used by animal breeders and farmers that we refer to as artificial selection. But the underlying genetic process does not differ from natural selection because 3 conditions are going to be satisfied. Also, artificial selection is going to be considered a special case of natural selection because it's basically going to be selected for or controlled by humans. Differential reproductive success is going to be determined, like we said, by the humans rather than nature. So in summary, animal breeders and farmers are making use of natural selection when they modify their animals and crops through selective breeding. They're basically breeding for the traits that they want to see. Since the differential reproductive success is determined by humans rather than by nature, this type of natural selection is also going to be referred to as artificial natural selection. Right, going into natural selection, and that can change the traits of a population in several ways. Here, you're going to have certain traits that are going to fall into easy categories. So some people have blue eyes, others are going to have brown eyes, and just as some tigers are going to have white fur, and then some are going to have orange fur. Other traits, such as height in humans, is going to be influenced by many genes and also environmental factors. So that's going to be a continuous range of phenotypes that's going to be occurring. So whether a given trait is going to be influenced by a gene or a complex interaction of many genes and the environment, it can be subject to natural selection and be changed in several ways. One way is going to be directional selection. That's going to be individuals with one form of the extreme trait in a range variation in a population will have the higher fitness. For example, milk production in cows is a good one. There's going to be a lot of variation in milk production from cow to cow. And so as you might expect, farmers are going to select for the cows that have the highest milk production for breeding. And once they've done this, they're going to do this over and over again. So the cows start producing more and more milk. So the results from this type of selection between like 1950 and 1945, average milk supply production is going to increase by about 50% in the United States. Those at the other end of the range, the cows that produce the smallest amounts of milk, are going to have a reduced fitness. So since these farmers don't allow them to reproduce and therefore contribute to less milk alleles, then that's going to be less likely to carry into the subsequent generations. Turkeys on poultry farms are going to have such large breast muscles that they can't get close enough to each other to mate. So how has this trait evolved if they can't naturally mate together? Well, what happened is hundreds of experiments in nature and in laboratories is going to demonstrate the power of directional selection. So one of these is going to be a laboratory selection of animal breeders that nearly any trait that they choose to exaggerate via directional selection responds dramatically, sometimes with these absurd results. So turkeys, for instance, are going to have been selected for a larger breast muscle, mainly because that's what most people prefer when they're eating turkey. So that's going to make them more valuable to farmers. This directional selection has been successful to the bird's breast muscle, but now that they're so large, it's impossible for them to mate. So all the large-scale turkey farms are going to actually have to go in vitro or artificially inseminate these turkeys. Stabilizing selection, so individuals with intermediate phenotypes are going to be the most fit. So you don't want one that's super tall or super short. It tends that the one in the center is going to be the most fit in some cases. So how much did you weigh at birth? Was it more than 10 pounds? More than 11? Was it close to 22? Well, unlike directional selection, for which the increased fitness at one extreme or the other, and then the one in the middle is going to be reduced, stabilizing selection is going to occur with individuals with an intermediate phenotype is going to be most fit. So the death rate among babies, for example, is going to be lowest in the 7 to 8 pound range. And that's going to rise above those who get heavier or for those who are smaller. Then we have what's referred to as a disruptive selection. These are going to be individuals with extreme phenotypes and where they experience the highest fitness, and those with intermediate phenotypes have the lowest fitness. So you see on both ends, but not the middle. So looking at this one, which graph illustrates the result of directional selection? That's going to be graph 1. So in summary, natural selection can change populations in several ways. Directional selection, in which the average value of the trait increases or decreases. Stabilizing selection, in which the average value of the trait remains the same while extreme versions of the trait are selected against. And disruptive selection, in which individuals with extreme phenotypes have the highest fitness. So we're going to go through that and skip that part. So starting with 817, natural selection can cause evolution of complex traits and behaviors. So with this one, in 1954, William Thompson is going to train a group of rats to run a maze for food. There's going to be a huge amount of variation in the rats' abilities. Some are going to be fast, some are going to be slow. So Thompson then selectively bred the fast learners, called maze-bright rats, with each other, and the slow learners, called maze-dull rats, with each other. Only after 6 generations, the maze-dull rats are going to make twice as many errors as the maze-bright rats before mastering the maze. Bright rats were going to develop at a solving complex mazes, and they're going to be a lot faster. So 50 years later, it's still going to be unclear which actual genes are going to be responsible for maze running behavior. Yet, the selection experiment still demonstrates that a strong genetic component to that behavior. And so here you see the little rats in their maze. How can a wing evolve if 1% of a wing doesn't help that organism fly or glide? Well, often a structure can appear because they serve some other purpose. So here we see a tiny wing nub, and in some it's going to help with thermoregulation. So it's going to help cool that animal or insect off. And so it's beneficial to have that nub. So when that nub size actually starts to increase and goes to the intermediate, it becomes more aerodynamic. So it started off trying to thermoregulate, trying to cool it off, and as it got bigger, it actually helped it be able to fly. So that's going to be referred to as a functional shift. So with this one, primordial insect wings were first used for blank. As size increased, they could be used for blank. The correct answer is going to be number 4. Evidence for evolution is going to be overwhelming. So we have fossil record documents that are going to show us how things have evolved over time. And this is going to give us 5 primary lines of evidence. We have the fossil record, which is physical evidence of organisms that have lived in the past. We have biogeography, and that's going to be patterns in geographic distribution of living organisms. We have comparative anatomy and embryology, and that's going to be the growth, development, and body structures of major groups of organisms. We have molecular biology. That's the examination of life at the level of individual molecules. And we have laboratory and field experiments. And that's basically the implementation of scientific method to observe the study of evolutionary mechanisms. So here you're going to see fossilized remains. And with these fossilized remains, you're able to actually see that it had specific bones. It's going to have soft tissue that left an outline. It looks like it had feathers. So number one, with fossil records, we can use what's called radiometric dating, and that's going to confirm the Earth is going to be very old, and it makes it possible to determine the age of the fossils. By analyzing fossils remains, paleontologists can then reconstruct what organisms looked like a long time ago. Then they can also use that to learn how organisms were related to each other and understand how groups of organisms evolved over time. So this shows how these isotopes in the organisms break down over time, and so they can use this half-life using that radiometric dating to determine the average age of that fossil. We can go through the evolutionary history of horses by looking at these fossil records. So because of this record, we know that since horses first appeared around 55 million years ago, there have been many branches off that evolutionary tree, a sample of which is shown here. Next, we have the Tiktaaliks. And so these are going to actually transition, and these fossils seem to represent the phase between lobed finned fish and terrestrial vertebrates. So you see how that lobed finned fish has the same bones or same bone structures. They're done by color. Then you see how it moved to the Tiktaalik, and then the early amphibian. So with the biogeography, that's the study we said of the distribution patterns of living organisms around the world. Patterns that Darwin's going to use to find relations between animals are still going to be looked at today. Species often more closely resemble species that live less than 100 miles away from them, but in radically different habitats than the species that live thousands of miles away in nearly identical habitats. For example, we have the Hawaiian honeycreeper. So here's the Hawaiian honeycreeper. And so when you look at these here, you have the habitat of the first one. It's going to be a wet forest, about 1,100 meters. You see what it's eating on. Then you're going to have the second one. It's going to be in the northwestern slopes. You see its diet. And then you go to the third one. It's the wet forests that are going to be above 1,250 feet or meters. So with these Hawaiian honeycreepers, they're going to resemble a common ancestor from the mainland North America, but all are going to have their unique features. All right, going into the next one, the history. What does it matter? Does it matter at all? Who arrived first? Well, when it comes to species distribution, this history is actually going to matter. Species are not going to be really distinguished from scratch to fill a particular niche. What's going to happen is whoever arrived first, usually a nearby species, is going to take up numerous different lifestyles in numerous different habitats, and the populations are going to ultimately adapt to their new environment, and they're going to evolve. So in the Hawaii, it appears that the finch-like descendant of the honeycreepers arrived around 4.5 million years ago and rapidly evolved into a large number of diverse species. The same process has also occurred and continues to occur in all locales, not just on islands. So here we see large isolated habitats also have interesting biogeographic patterns. So the Australian and Madagascar are going to be both filled with unique organisms that are going to be clearly not closely related to organisms elsewhere. For example, in Australia, The marsupial species, rather than placental mammals, fill all of these usual roles. There are marsupial wolves, marsupial mice, marsupial squirrels, and marsupial anteaters. They're physically going to resemble the placental counterparts for the most traits, but the molecular analysis shows that they're actually more closely related to each other, sharing a common marsupial ancestor. And so their relatedness to each other is also going to be revealed by similarities in their reproduction. Females all give birth to offspring at a relatively early state in development, and then that offspring will finish their development in a pouch. So if you observe any vertebrate embryos, it all passes through the same type of stage in which the little gill pouches on the sides of the neck will form. It will also pass through a stage in which it possesses a long bony tail. This is going to be true whether it's a human embryo or that of a turtle or a chicken or even a shark. These gill pouches are going to disappear before birth in all but the fishes. And so similarly, we also don't see tails in humans, but we could see them in other animals. Such common embryological stages indicate that organisms share a common ancestor from which they have all been modified. So the study of these developmental structures and the adult body forms of organisms helps provide our third line of evidence, helping us to see the evolution occurs and to better understand the process. Now going into homologous structures. Among adults and several features of anatomy, they're going to reveal that there are specific structures that will match amongst different animals. And so we refer to this as a type of relatedness. So notice how the human arm has a humerus, the horse does, the bat, and the porpoise. Then we move on to the forearm bones. We have the radius and ulna in the human. You have one in the horse, you have the same type of bone in the bat, and then in the porpoise. And then you go down to the carpals and your metacarpals. So comparative anatomy is going at the extreme is going to be a homologous structure that sometimes comes to have little or no function at all. Such evolutionary leftovers are called vestigial structures, and they exist because they had value ancestrally, but now they do not. So some vestigial structures in mammals are going to include things such as molars that continue to grow in vampire bats, with eye sockets with no eyes in some populations of cave-dwelling fish, and pelvic bones in whales. Even humans have vestigial organs. Take, for example, the appendix. In other primates, it's going to be an organ where fermentation occurs with plant products, and it helps break it down. In humans, we don't really use that structure anymore. You can remove it and you will be fine. It does cause lots of problems sometimes. It will get bacteria in it and can become inflamed, and then it can burst and actually lead to other problems. Here you're going to see the vampire bat, and you see it has molars towards the back of the teeth. It doesn't need these molars anymore because it's not grinding food. Next, we go on to convergent evolution. This is going to create what we know as analogous structures. These all develop from different original structures, and they're going to serve the same purpose. So with this one, which line of evidence for evolution notes similarities between vertebrate embryos? Number 2 is correct. Molecular biology is going to reveal that common genetic sequences are going to link all life forms. Basically, we're all made up with the same A's, T's, C's, and G's. And for the most part, they're going to be put together somewhat similar. For example, if you're making a protein to code for hair or fur, they might be very similar. So with these similarities, they give us related versus unrelated individuals. They show how the closer individuals are going to be related, the greater the proportion that their DNA is the same. And the more distantly you are to another individual, the more differences you'll have in your DNA. So compare the DNA sequences for individual genes. For example, in rhesus monkeys, we find that there's 138 amino acids that are the same as those that are found in the human hemoglobin. Both humans and the rhesus monkeys have a total of 146 amino acids that code for the beta chain. There's only going to be 12 differences between humans and these monkeys. Recency of common ancestry. We can help estimate the evolutionary relatedness made from comparative anatomy, embryology, and fossil records. We also have what's referred to as molecular clocks. So the longer two species have been evolving on their own, the greater the number of changes in that amino acid sequence. So we can go back and see that that's further back on the molecular clock. So looking at this one, it says, based on the difference in the amino acid sequence of beta hemoglobin chain, to which animal listed below are humans more closely related? The correct answer is going to be the rhesus monkey. When did humans and birds last share a common ancestor? That is going to be number 3. And looking at this one, I'll let you read through it. And the correct answer is 1. Okay. Laboratory and field experiments are going to enable us to watch evolutionary progress. So a fifth line of evidence is going to be the occurrence that comes from multi-generation experiments and observations. So we can look at this with changes in frequencies of various alleles. So we're going to study golf course grass. All grass was the same type. Rough was almost never cut, the fairway was cut occasionally, and the putting greens cut frequently. So over several years, the grass changed quickly. The putting greens died out quickly, they had a high reproductive rate and had a high seed output. The fairway is going to have a slower reproductive rate, and the rough is going to have the slowest reproductive output and rate. So looking here, we can see the changes. So over time, it's going to develop into 3 distinct species depending on how it was cut and how frequently. Evolution of antibiotic resistance occurred. So in the 1940s, we were going to first start using antibiotics when it was discovered that mold on a petri dish actually killed the bacteria around it. So when we first used this medicine, penicillin was uniformly effective in killing Bacterium Staphylococcus aureus. And so you see that clearing, that kill zone, that zone of inhibition. Today, however, natural selection has led to an increase in antibiotic-resistant alleles, and humans are increasingly at risk for an untreatable Staphylococcus infection. We also saw that with the fruit flies when we were able to change them from living about 20 hours to 160 hours. So in summary, replicated controlled laboratory selection experiments and long-term field studies of natural populations allow us to watch and measure evolution as it occurs. That's going to finish us up for Unit 8.